Bioinfocongress V Doktor Eda Yıldırım X kromozomunun dozajının dengelenmesi, mekanizmaları ve kansere etkileri. Doktor Eda Yıldırım, 1997 yılında ODTÜ Fen Fakültesi Biyoloji bölümünden mezun olmuştur. Doktor Yıldırım, doktora çalışmalarını TRPC ve IP3 reseptörlerinin hücresel ve patofizyolojik rolleri üzerine Kaliforniya Üniversitesi, Los Angeles'ta Dr. Luz Birnbaumer'ın laboratuvarında yapmıştır. 2005 yılında doktora sonrası çalışmalarına başlamak üzere Harvard Tıp Fakültesi, Massachusetts General Hospital Moleküler Biyoloji bölümünde bulunan Dr. Jenny Lee'nin laboratuvarına katılmıştır. Bu çalışmalar sırasında Axis kodlamayan RNA'sının X kromozomunun inaktivasyonunun sürerliliği ve kansere olan etkilerini araştırmıştır. Yaptığı çalışmalar, Axis RNA'sının X kromozomunun inaktivasyonunun sürerliliği için gerekli olduğunu ve fare sisteminde dişilere özgü kan kanserlerinde baskılayıcı fonksiyonu olduğunu göstermiştir. Dr. Yıldırım, 2014 yılında Duke Üniversitesi Tıp Fakültesi'nde kurduğu laboratuvarında memelin sistemindeki kodlamayan RNA ve hücre çekirdeğinin yapısı tarafından kontrol edilen epigenetik mekanizmaların gen regülasyonuna ve genom fonksiyonuna etkilerini incelemekte, aynı zamanda bu mekanizmaların gelişim ve kanser için önemleri konularında çalışmalar yapmaktadır. Dr. Yıldırım, 2018 yılında National Academy of Sciences tarafından Kavli Frontiers of Science Fellow olarak seçilmiş, 2014 yılında ise Duke Üniversitesi Tıp Fakültesi tarafından verilen Whitehead Scholar ödülünü almıştır. Dr. Yıldırım, European Youth Search Council, National Science Foundation ve National Health Institute tarafından organize edilen proje değerlendirme kurullarında görev yapmaktadır. Ağustos 2014 yılından bu yana Duke Üniversitesi Tıp Fakültesi Hücre Biyolojisi bölümünde yardımcı doçent ve epigenetik programının direktörü olarak görev yapmaktadır.
Sayın Doktor Eda Yıldırım ve değerli katılımcılarımız, Bioinfocongress V etkinliğimize hepiniz hoş geldiniz. Değerli hocamız bizlere bugün X kromozomunun dozajının dengelenmesi, mekanizmaları ve kansere etkileri konulu bir konuşma gerçekleştirecektir. Konuşma bitiminde ekibimizin hazırladığı ve katılımcılarımızdan gelen sorulardan oluşan soru-cevap bölümümüz olacaktır. Merhaba Eda Hocam, tekrar hoş geldiniz.
Merhaba, çok teşekkür ederim davetiniz ve bu güzel organizasyona beni de eklediğiniz için çok teşekkürler.
Biz teşekkür ederiz hocam. Dilerseniz sunumuza başlayabilirsiniz.
Tabii ki, çok teşekkür ederim. Tekrar sunumuma bu güzel organizasyonu bu anlamlı günde gerçekleştirdiğiniz ve bizleri davet ettiğiniz için çok teşekkür ederim. Hepimizin Cumhuriyet Bayramı kutlu olsun diyorum. Ön konuşmadan da belirtildiği gibi şu anda çalışmalarıma yardımcı doçent olarak Düzce Üniversitesi Tıp Fakültesi'nde hücre biyolojisi bölümünde devam ediyorum. Çalışmalarımız daha çok X kromozomunun dozajının dengelenmesindeki etkili mekanizmalar, bu mekanizmaların kansere etkileri ve ayrıca nükleer por kompleksinin gen regülasyonu ve genom fonksiyonu üzerinde yoğunlaşıyor. Müsaade ederseniz konuşmamın geri kalanını İngilizce olarak devam edeceğim. Ama sorularınızda konuşmalarımıza Türkçe olarak devam edebiliriz. In various organisms, development of genetic sex determination leads to gene dosage imbalances between sex chromosomes and autosomes and between the sexes. In mammals, dosage compensation occurs at two levels, both of which target the X chromosome. One of these processes is a developmentally regulated process called X chromosome inactivation, which ensures equal dosage of X-linked genes between the females and males. In the developing female blastocyst, the epiblast cells acquire two active X chromosomes, or XAs, one of which is randomly chosen to be the future inactive X, or as I would refer to it as the XI. Initiation of silencing on the inactive X chromosome depends on the expression of a long non-coding RNA transcript called EXIST, which codes the inactive X chromosome, and together with DNA methylation ensures chromosome-wide silencing. While X chromosome inactivation silences one of the X chromosome, it creates a dosage balance in the active X chromosome, since there is only a single copy of the active X and two doses of autosomes. And this process is thought to be balanced through another dosage compensation process called XA hyperactivation. It's well known that the initiation of X inactivation depends on EXIST expression. However, even though EXIST is expressed continuously during the lifetime of the female organism, it has been thought to be dispensable for the maintenance of X inactivation. Despite this fact, loss of EXIST expression, the presence of supernumerary X chromosomes have long been associated with various human cancers, including breast, ovarian, and blood cancers, and testicular germ cell tumors in males with additional X chromosome, bringing up the question of whether there's a causal link between the X chromosome dosage and cancer progression. And I was interested in addressing this question during development. And for that purpose, I utilized a conditional EXIST knockout mouse that was developed in Rudolf Jenisch's lab at MIT. And I crossed these female animals to a transgenic mouse in which the Cre recombinase is under the control of VAV1 gene. And VAV1 is specifically expressed during the embryonic day 10.5 in the hematopoietic stem cells, which give rise to the multipotent and committed progenitors of the blood lineage, as well as the mature cells. Therefore, we were able to delete EXIST during the hematopoietic lineage, beginning with the start of stem cells towards the mature cell lineage. And this was important because as initiation happens in the second and 2 to 4 stage cell stage during development, maintenance happens after embryonic day 4.5. Therefore, the hematopoietic lineage development falls into the maintenance stage. So we could study the stages of X inactivation and how EXIST has a role in the maintenance process using this cell type. So this work was published several years ago, but I would like to give you a brief summary of our findings. We first collected spleen cells and ensured that exist RNA was successfully deleted by performing sequential RNA and DNA FISH using probes that interact with the exist locus. And as you can see, compared to the control cells, there was no exist expression in the mutants. We next investigated how exist loss impacted the survival of the female and male mice. And as you can see from this survival curve, we only detected lethality in the exist homozygous and heterozygous female mutants compared to their wild-type counterparts, as well as the male mutants or the male control mice. The lethality that associated with the female mutants also presented certain signs of disease that included the abdominal enlargement, as you can see in the exist female mutants, and the tumors that were even evident to the eye from the external examination of these mice. So together with two pathologists, James Kirby and Diane Brown, we began to investigate the pathological characteristics of the exist mutant animals by investigating how the bone marrow, peripheral blood, spleen, lymph nodes, and the secondary organs such as the kidney, liver, and lungs were affected due to exist loss. So the first phenotype that we observed was the female-specific spleen enlargement, or splenomegaly, as we call it. This was evident with the almost 4 to 6-fold increase in the spleen to body weight ratio of the exist mutant females. And when we took cross sections of the spleen, the expected anatomy of the spleen was completely distorted, whereby the germinal centers where B and T lymphocytes reside, as well as the red pulp where you find megakaryocytes, myeloid cells, and erythrocytes were all enlarged. And this became even worse with the progression of the disease when we examine the exist female mutants. And I am presenting you Ki67 stain slides. This is a marker of proliferation. And as you can see, there is massive proliferation. And as I'm not showing you, but you can go back to the reference and check the stains we used for specific cell lineages, we detected proliferation in all three major lineages, including myeloid, lymphoid, and erythroid cells. And therefore, we concluded that exist loss leads to hyperproliferation in the mature cells of the hematopoietic system. This phenotype also suggested that there is likely a bone marrow dysfunction, given the fact that the bone marrow is the source for the expansion of the hematopoietic cells. So in order to check whether there is a bone marrow dysfunction, we took sections of the bone, as you can see here, from different stages of mice, so we can see the progression of the disease. And we compared the wild type to mutant mice at 2, 4, and 11 months and examined the cellularity changes as well as the presence of reticulin fibers, which is an indication of bone marrow dysfunction. And as you can see in the mutant mice, we detected loss of cellularity, and this was later described as these mice being anemic, as I will share with you later. And there were the presence of reticulin fibers, which became really intense and severe as these mice aged. So we concluded that exist deletion leads to bone marrow dysfunction in the form of myelofibrosis. With Diane Brown, we also examined the bone marrow aspirates, which were collected from wild type and exist mutant mice. And compared to the wild-type cells, we detected major myeloid lineage hyperproliferation, and within these cells, we detected several cells that showed abnormal mitotic figures with chromosome abnormalities, suggesting that there are certain factors that contributed to the genomic instability in these mice. Next, we looked at the peripheral blood to see if there were any multilineage defects in the circulating leukocytes. So in a wild-type blood smear, what you expect to find are the platelets and few lymphocytes that are residing naturally. But in the exist mutant mice, the peripheral blood presented signs of leukemia with increased white blood cell counts up to almost 10 to 15-fold. Therefore, we concluded that there were multilineage defects in circulating leukocytes, which also presented signs of especially chronic myelomonocytic leukemia. So another feature of these animals was that we, because there was a bone marrow dysfunction, there was massive proliferation of hematopoietic cells elsewhere in other organs, including the liver, the spine, lung, and kidney. And here I am showing you the solid tumors that exist in various tissues. And these tumors contained multinucleated giant cells, which stand for histiocytic cell markers, suggesting that these metastases might be due to the histiocytic sarcoma in these animals. And we are currently carrying out experiments to look into this type of cancer in these mice. So with these characteristics, including the myeloproliferation, myelodysplasia with the presence of atypical and increased leukemia, which was representative of myelodysplasia, and the myelofibrosis, these characteristics resembled a cancer that is also observed in human population called myeloproliferative neoplasm and myelodysplastic syndrome. And this phenotype was 100% penetrant in all female mice that we examined that were exist mutant. And in 30% of our cohort, we also detected leukemia in the form of chronic myelomonocytic leukemia and detected undifferentiated and immature erythroid cells, which was an indication of erythroleukemia-like syndrome, as well as the presence of solid tumors, which suggested symptoms of histiocytic sarcoma. So this was my postdoctoral work back in 2013, and we were very excited to understand the origins of this disease with the collaboration with David Skadden's lab at Harvard. We also performed bone marrow transplantations in order to show the cell of origin of this cancer phenotype. These experiments were quite straightforward, in which we collected bone marrow cells from the mutant donors and transplanted them into wild-type recipients, which were lethally irradiated. So we can see whether the cancer phenotype that we detect in exist mutants are transplantable, and we can recapitulate the phenotype in these recipients. And indeed, within 3 months of transplantation, we were able to detect the leukemia phenotype with increased white blood cell counts, and we can see enlargement of the liver, as we detect in exist mutants. And in the wild-type recipients, we can see splenomegaly that also presented the similar anatomical and pathological changes we observe in exist mutants. And we also detected the solid tumors with the help of H&E staining. And with reticulin staining, we can recapitulate also the bone marrow fibrosis in the recipients. And all these results suggested that exist-dependent cancers are hematopoietic in origin. So after starting my lab at Duke with my graduate student Tianqi Yang, we decided that we would like to look into the cellular and the molecular defects that associate with the exist-dependent blood cancers in female mice. So just to give you a brief introduction and a background on the hematopoietic lineage. This lineage is very well characterized, which makes it very, very applicable for in vitro and in vivo studies, and therefore allowed us to study the lineage at the stem cell level, which we call the LSK plus stem cells, that contain the long-term hematopoietic stem cells, short-term hematopoietic stem cells, and the multipotent progenitors, and the LSK minus progenitors, which include the common progenitors, which are committed progenitors, as well as the mature cells. So during my talk, I will discuss the LSK plus stem cells, LSK minus progenitors, and the Lin minus cell type is the combination of the LSK plus and LSK minus populations, and they devoid of the mature cells of the hematopoietic lineage. And as I indicated, these cells are very well characterized, especially for the expression of their cell surface markers. Therefore, we can utilize FACS to sort and detect the quantity and quality of these cells. So, and for some of these markers include c-KIT or SCA, which allows us to distinguish the stem cell population from the progenitor population. And using CD48 and CD150, we can further subgroup the stem cell population into 4 and distinguish the multipotent progenitors, such as the HPC1 and HPC2, and the hematopoietic stem cell group. And we can use CD34 and Fc gamma receptor to distinguish the committed progenitors, including the megakaryocyte erythrocyte progenitors, or MEPs, common myeloid progenitors, or CMPs, and the granulocyte macrophage progenitors, called GMPs. And using IL-7 receptor alpha, we can also distinguish the common lymphoid progenitors. So using this technique, which is a combination of staining cells for the cell surface markers and then performing FACS, Tianqi was able to investigate how the stem cell and the progenitor cell population change upon exist loss. So as you can see in this, in these graphs where we quantify the stem cells and the progenitors in percentage and cell number, we can see a significant, actually up to 4-fold increase in the stem cell population in exist mutant mice. And if we look at the subgroups of the LSK plus stem cells, this expansion was significantly apparent in the HPC1 and HPC2 population. And this brought up the fact that the exist loss is causing an expansion in the stem cell compartment. We also looked at the progenitor subpopulations, as shown here, and we didn't see a significant change in the committed progenitors, except the common lymphoid progenitors, which I will come back to later. So even though we didn't detect a change in the number of progenitor cells, when we examine the signature genes that make up the committed progenitors, we detected a major decrease in the CMP signature genes as well as the MEP signature genes, while we see an increase in the GMP signature genes in exist mutants. So this argued that exist deletion facilitates the lymphoid progenitor differentiation, as I showed you earlier, but hinders myeloid progenitor differentiation towards the megakaryocyte and erythrocyte lineages, as it was evident from our cellular and pathological analyses in our earlier publication. And this work was, just to tell you, was published last year. So I would advise that if you have any need to look into details, please check our publication. So we next were interested in the reason for the expansion of stem cell population in the X mutant mice. So as you might already know, cell cycle is a critical determinant of hematopoietic stem cell self-renewal and differentiation. The hematopoietic stem cells present quiescent phenotype in the bone marrow, and they only enter cell cycle when there's a need for differentiation and production of other cell lineages of the blood, or self-renew so that they can repopulate the pool of stem cells. So we wanted to take advantage of these characteristics and understand whether cell cycle contributed to the expansion of the LSK plus stem cells. And we utilized a technique called Ki67 based FACS analyses. And in this case, Ki67 is used as a marker to distinguish between the cycling or G1 or the quiescent or GO cells. So if you focus on the wild type bone marrow cells here, where we separated the LSK plus and LSK minus cells, you can see that almost 92% of the LSK plus cells are at the GO stage, while only 7% are cycling. But in the progenitor population, this is almost equally distributed. When we look at the X mutants, this ratio was completely distorted, whereby we see more cells, almost 3 to 4-fold more cells cycling in the bone marrow, and there were less cells that are at the quiescent state in the stem cell population. So Tianqi also quantified this across different cell groups. And as I've shown you earlier, there was a major decrease in the quiescent cell percentage in the stem cell population. And this was also apparent when we look at the cycling cells, as you can see here. So we concluded that X deletion leads to an impaired quiescence in the LSK plus stem cells. Next, we wanted to understand how specific this effect was at the cell cycle stages. And for that, in order to basically prevent any signals that might interfere with the cell cycle process, we decided to isolate the bone marrow cells from 3 months old wild type and X mutant mice and culture them in the presence of growth factors for 2 days. And we call these cells as post-culture cells. And in order to look at the S phase specifically, we treated the cells with EDU and allowed the cells to be in this culture for 1 hour. And we utilized these cells to perform exist RNA FISH to distinguish the cells that lack exist and contain exist. I forgot to say we use heterozygous animals for this experiment, which allowed us to look both the wild type cells as well as the mutant cells. So, and we used the click-it reaction to detect the EDU incorporation, and we also utilized a histone modification called histone 3 lysine 27 trimethylation to detect for the chromatin-specific changes in the X mutant mice. In parallel to this experiment, we performed exist RNA FISH together with K27 trimethylation immunofluorescence, and we added a marker of mitosis, which is histone 3 serine 10 phosphorylation. And this is something that an earlier talk referred to by Nurhan Özlü in the morning. So there are specific histones that associate with certain histone modifications, and H3 serine 10 phosphorylation is one that is used as a marker. So this on the left, the panel shows you our staining, basically order and results. We were able to stain exist, and as you can see, exist forms a cloud on the X chromosome territory, and this is overlapped with the histone modification, histone 3 lysine 27 trimethylation, and we were able to detect also EDU with the ClickIt assay, and this way we can distinguish exist positive and K27 positive cells, and we can distinguish EDU positive and EDU negative cells. And as expected, since we were using heterozygous animals, we were able to detect half of the cells we looked at contained exist and half didn't. And among these cells, the exist and H3K27 minus cells, which are marked in red, were the ones that had higher percentage of EDU positive cells, meaning that exist loss leads to increased proliferation, because we can see that that there are more cells at the S phase when they are exist mutant. And this was also true for the histone 3 serine 10 phosphorylated cells. We can see cells that are positive for this mark, suggestive of the increased proliferation in LSK plus cells. So the next question we asked was that, are there molecular mechanisms that link exin activation maintenance to genome function in female cells? So for this, we turn to RNA-seq analyses, whereby we examine 3 cell populations, including the LSK plus stem cells, LSK minus progenitors, and the lineage negative cells, which contain these 2 populations. And we perform CDF cumulative fraction plots, where we look at the global expression of X-linked genes and compare it to the autosomal genes. And a right shift in the curve would suggest there is more gene expression on the given chromosome or chromosomes. And as you can see that in the stem cell population, we can see a significant right shift with the p-value, and this change became even more significant in the lineage cells because it contains more differentiated and mature cells of the multipotent progenitors. But we didn't see such difference in the LSK minus cells or the progenitors. So this result suggested that the exist loss results in upregulation of X-linked genes, and exist is critical for the expression of or control of the maintenance of X-linked gene expression. Next, we perform gene set enrichment analyses, or GSEA, to look what kinds of genes are upregulated. And we see several categories in the stem cell compartment which associate with cell cycle associated genes. And these included sister chromatid segregation, sister chromatid cohesion, and there were some immune response related gene groups that associate with type 1 interferon and defense response to virus. We didn't see enrichment of these genes in the progenitor population. So we further looked what kinds of genes were within these categories and how they differed between stem and progenitor cells. And we can see, as I listed here, and these are all X-linked differentially expressed genes, or DEGs, we can see a major upregulation of a majority of the cell cycle related genes that are listed here to be upregulated in the stem cell compartment. And this was also true for the immune-related genes that are listed here. So this suggests that the changes due to exist loss on the X-linked gene upregulation is cell type specific, and depending on these changes and the types of genes that show differences in X-linked gene expression, this severity of the phenotype would be affected. So we next want to see what are the common differentially expressed genes between different cell populations. So here I'm showing you two Venn diagrams, which is representative of upregulated differentially expressed genes, or DEGs, on the X chromosome and across the autosomes. And for the X chromosome, we detected almost 30 genes to be upregulated that were common between the three populations, and this constituted 13% of the differentially expressed genes. And Tianqi did something good, I think, when she took all these 30 genes, which are listed on the right panel in the heat map, and she looked how the expression of these common differentially expressed genes change in other tissues in which exist was deleted by us or by other groups in the field. And these tissues included brain, certain subsections of the gut, including the crypt, the polyp, and the kidney. And as you can see, several of them were upregulated across the board, as well as in the bone marrow, which suggested that actually this effect that we see, which is the upregulation of certain X-linked genes, was specific to exist, because this is also true for the other tissues. Interestingly, when we examine the characteristics of these genes, such as DDX3X, KDM6A, KDM5C, these genes represent the genes that continue to be biallelically expressed, meaning from both of the X chromosomes upon X chromosome inactivation. So, and they're called escape genes. And the best example is exist, because even though the inactive X chromosome is silenced, exist expression continues to be expressed on the XI. Therefore, it escapes the inactivation process. So unfortunately, we weren't able to look at the allelic nature of the transcriptional changes using our bone marrow cells because we weren't able to distinguish the two alleles of the X chromosome or X-linked genes based on strain-dependent single nucleotide polymorphisms. So for that reason, we turned into mouse embryonic fibroblasts that were isolated from the exist conditional knockout mouse, and whereby they were transformed with the SV40, and they were transfected with the Cre recombinase to delete exist, and they were cloned, so we can detect deletion of exist specifically on the XI and ask inactive X chromosome specific questions and study the differences in the expression of X-linked genes, whether it comes from the XI or the XA. And this is possible because these mouse embryonic fibroblasts have hybrid origin. So half of their genome is mus musculus and half of their genome is mus castaneus. These are two different strains of mice. So, and these were first described in 1997 in a Cell paper by my former postdoc advisor, Jeannie Lee's group. So she kindly shared these cells with us, and we utilized them to perform RNA-seq experiment and looked at the allelic expression of transcription, and we, depending on the threshold we set, we were able to define exist-dependent genes and exist-independent genes on the X chromosome. So if they are exist-dependent, they will show a change in their expression level, and if they are exist-independent, they would not show such change. So we categorize the genes into escape genes and exist subjective genes, and escape and subjective genes, the difference between the two is escape genes would be expected to be expressed from both alleles, and the subjective genes are the ones that are expressed monoallelically. So among the escape genes that we defined in the wild type, we detected 14 of them to be exist-dependent and 3 of them to be exist-independent, while among the subjective genes, we detected 1 of them to be exist-dependent. So we later utilized these gene groups to ask the question of how X-linked gene expression overall changed. But before I get into the details of those analyses, I would like to give you a brief background on the stages of X inactivation. So this is a hierarchical event, and when before the initiation starts, RNA polymerase II is engaged with the active X chromosome, and exist expression is suppressed, and there is demethylation of the X chromosome, and with the initiation of X inactivation and with upregulation of exist, several chromatin regulatory factors and modifiers get recruited to the X chromosome, leading to the histone 3 lysine 27 trimethylation, which is catalyzed by polycomb repressive complex, or EZH2 component, and there is ubiquitination through PRC1, and several factors such as yin-yang-1 factor, or YY1, that gets recruited, which is a transcription factor that can help with the nucleation of exist on the X chromosome. And this process is followed by the propagation of silencing and maintenance across the X chromosome, and with the recruitment of DNA methyltransferase 1, there is high function of DNA methyltransferase 1 for DNA methylation, which locks in the silent chromatin state. So the first suspect that we went after to look that might be, responsible for the upregulation of X-linked genes was the depletion of H3K27 trimethylation, because this is the major mark that helps with the silencing on the X chromosome. So Tianqi took Lin- bone marrow cells from wild type and exist heterozygous mutants, and she prepared chromosome spreads. So basically, the dots that you're detecting or you're seeing here are individual chromosomes. And then she performed DNA fish using XPaint, which labels the X chromosome, and did immunofluorescence against histone 3 lysine 27 trimethylation to label the inactive X chromosome, as you can see here. So, as represented in this figure, we see a complete loss of the H3K27 trimethyl through immunofluorescence. Of course, this technique is somewhat subjective. Therefore, we also performed chromatin immunoprecipitation to detect histone 3 27 lysine 27 trimethylation across the genome using H3K27 trimethyl antibody. And we perform bioinformatic analyses to look at the changes or the occupancy of this mark across genes. So just to give you an example, if you're not familiar on this kind of data set, we took the transcription start site as a reference point, and TES is the transcription end site. And then we took 3 kb downstream of the transcription end site and 3 kb upstream of the transcription start sites. And we align all the reads that come from the sequencing of the chromatin immunoprecipitation data. So, and basically these signals that you're detecting here are showing you the reads that associate with specific regions in the genome, and they are additive. So the blue line represents wild type and red line represents exist mutant. And we categorize this data set into upregulated and downregulated differentially expressed genes and use the active non-DEGs as a control group. So you can see that across the board, which did not depend on the upregulation or the downregulation of the DEGs, we see a decrease in the K27 trimethylation, independent of the expression status of X-linked genes in exist mutants. So this suggested that indeed there is an effect of exist on the K27 trimethylation, but it's not the exclusive signal that causes the transcriptional upregulation of the genes that we detected. So we next looked into whether the chromatin accessibility at these sites mattered because an earlier publication suggested that exist loss causes increased chromatin accessibility. So we utilized the LSK plus stem cells, which we sorted from the bone marrow, and we performed a technique called attack-seq. And attack peaks basically represent the sites where you have increased chromatin accessibility, or the chromatin is accessible at those sites. So here, these bars present you the upregulated common DEGs, lineage-specific DEGs, and downregulated DEGs, as well as our control group, which is the active non-DEGs. So you can see that within the transcription start site proximal locations of the peaks versus the distal sites, there is a major increase in the attack peaks or chromatin accessibility at the upregulated DEGs compared to the lineage-specific DEGs or the downregulated DEGs. And this was also true for the TSS distal sites for the upregulated DEGs. And here I'm presenting you on the right few upregulated DEGs and their attack profile, where you can see an increase in the mutant. So next, we wanted to see in MEFs, because we can identify what happens in the inactive and the active X chromosome. We analyzed the K27 trimethyl chip data or chromatin immunoprecipitation in MEFs. And we divided the data into the TSS proximal and gene body specific. And what you can see is that across the board, including the exist-dependent and exist-independent genes, which was evident from also the bone marrow data set, there is a significant decrease in H3K27 on the inactive X chromosome. This is what the D score means. So the positive signal represents a signal from the inactive X chromosome, and the negative signal is the active. Or the loss of the signal from the inactive X chromosome. And if you look at the gene body, this was also true for the gene body. So loss of exist cause K27 loss both at the TSS proximal sites and the gene body in both exist-independent and exist-dependent genes. What about the chromatin accessibility in the MEFs? Similar to what we observed in the bone marrow cells, we can also detect chromatin accessibility loss or increase in the chromatin accessibility in only the exist-dependent genes, unlike the exist-independent genes, suggesting that this is a exist-dependent event. So next, we wanted to understand what are the kinds of motifs that we can detect where the chromatin accessibility increased in exist mutant mice. And for this, we turned to Homer analyses, where we looked at the transcription factor motifs that are enriched within the X-linked attack peaks in the MEFs and also in bone marrow cells. So at the top, I'm showing you the MEF data, and at the bottom, I'm showing you the bone marrow data. And when we look at the transcription factor motifs in both data sets, one transcription factor popped up to be significantly enriched for its motif, and which is yin-yang 1 or YY1. So as I introduced earlier, YY1 is a factor that has been previously identified by Yesu John in Jeannie Lee's lab as a factor for exist nucleation. And it has also been identified as a factor that helps with the chromatin architecture through which it controls the enhancer chromatin promoter interactions. So in addition to these roles, YY1 has a role in controlling transcription, either through activation or silencing of gene expression, depending on the context of chromatin. So it's quite of like a multifunctional protein, which made it a bit difficult to characterize on what it is doing during maintenance. So Tianqi performed a CHIP or chromatin immunoprecipitation against YY1 to understand where it is enriched and how it is changing in the absence of exist across the X or across the autosomes. And here I'm showing you how YY1 binding changes at the TSS distal or TSS proximal sites. Even though we didn't see a major change in the number of peaks that we detect, we see an increase in the YY1 binding, especially at the X chromosome distal sites compared to the autosomes. In lean cells or in bone marrow cells, also in the MEFs. And in order to see what kind of regions or cis-regulatory elements the distal sites might correspond to for YY1, we also performed a histone 3 lysine 27 acetylation chip, which is telling us the sites for active enhancers. And as you can see that there was a good correlation of enrichment of the H3 lysine 27 acetylation and YY1 binding. And when we overlapped these data sets, there was a good correlation and positive correlation between the two, meaning that exist loss leads to increased YY1 binding, especially at cis-regulatory elements, specifically at the distal regions or active enhancers, which might affect the chromatin organization across the genome, especially on the X chromosome. So with that, we also looked how YY1 binding changed at the common differentially expressed and lineage-specific differentially expressed genes in the negative and also MEF cells. So you can see that the increased YY1 binding was specifically affecting the common DEGs, or the ones that include the escape genes. And this was if we look at the MEF cells, this was also true for the XCI subjective genes, especially for the MEFs. And when we depleted YY1, unfortunately, we could not see like a straightforward answer for whether there is up or down regulation in the X-linked gene expression, mainly because of the dual effect of YY1 and X-linked gene expression. So at the enhancer sites, though, we see a major increase in the YY1 binding, both at the exist-dependent and exist-independent genes. So it seems that the YY1 effect is specific for the promoter sites, but a bit non-specific at the enhancer regions in this case. So our working model right now suggests that there are both exist-dependent and exist-independent silencing mechanisms during XCI maintenance. And for the escape genes, which are the ones that we think are more prone to exist-dependent upregulation in the absence of exist, because of the increased chromatin accessibility and the active transcription at these sites. And we think that YY1 helps with the promoter enhancer interactions within, at these genes. And that's something we are actively investigating using genome-wide and immunofluorescence type experiments. For the XCI subjective genes, these genes have low susceptibility to transcriptional upregulation upon exist loss. And we think that, again, the YY1 has a role, likely has a role either up or down regulation of the X-linked genes. And through some ChIA-PET-based assays, we're trying to investigate which genes are positively or negatively affected by YY1. And we're looking into other transcription factors that might be involved in this process. So we concluded that there are several factors that influence exist deficiency phenotype, and these depend on the cell type specificity in the variability of the XCI escape genes, which show tissue-specific expression, the accessibility of transcription factors, and their presence or absence in a given cell type, and the cell turnover rate. I didn't discuss this earlier, but the hematopoietic cells have a high turnover rate compared to other cell types like neurons, which do not necessarily divide. And we believe that the reason that we are seeing such a strong phenotype in the hematopoietic lineage is likely due to their self-renewal and high proliferation rate. So just to summarize the work I have just presented to you, we concluded that exist deletion leads to a preferential transcriptional upregulation of XCI escape genes. And we found that exist is a negative regulator of YY1 binding on the inactive X chromosome by restricting chromatin accessibility. And our data suggests that exist loss leads to lineage-specific differentiation and cell cycle defects. And we're actively investigating the nature of these defects at the moment. So with that, I thank you for your time and for being here today. I would like to acknowledge my lab members, especially Tianqi Yang, who is now a postdoc at Yale working on spatial transcriptomics. She spearheaded this project, and with her diligence, she did a wonderful job and a comprehensive study. And Shin Kadota and Yuming Xing, who helped her during the process. And Mackenzie, Kyra, and Maria are current lab members who are continuing the work that Tianqi started. And our collaborators, Jianhong Wu, who helped us with the bioinformatics analyses, and several cores at Duke who helped us with cell sorting and sequencing. Yaru Diao's lab, who helped us with the attack experiments. Stefan Pinter and Felix Krugel for analyzing the allelic data set, and our funding at Duke, Leukemia Research Foundation, and NIH. With that, I'll be happy to answer your questions. Thank you.
Çok teşekkür ederiz hocam. Dilerseniz ekibimizin hazırladığı ve katılımcılarımızdan gelen sorulardan oluşan soru-cevap bölümümüze geçebiliriz.
Memnuniyetle.
İlk sorumuza başlıyorum o halde. Daha önceki çalışmalar X'siz silinmesinin MIF'lerde XSI kaçış genlerini barındıran bölgelerde TAD benzeri yapıların yeniden kurulmasına neden olduğunu bildirmişti. Bu yeniden düzenlemenin olası etkileri nelerdir?
Güzel bir soru. Öncelikle TAD'nin ne olduğunu anlatayım. Bunlar Topologically Associated Domain olarak belirtiliyor. Ve kromatinin oluşturduğu hub'lar aslında. Yani kromatinler tad lokasyonları çevresinde bir araya geliyor ve enhancer promoter bağlantıları da bu tadlar sayesinde dengeleniyor diye biliniyor. Şu anda tadların aslında X inaktivasyonuna nasıl etkileri olduğu hakkında çok fazla bilgimiz yok ne yazık ki. Yalnız şunu biliyoruz ki tadların bağlandığı bölgelerde özellikle CTCF denen insülatör proteinleri çok yoğun bir şekilde bulunuyor. Ve soruyu soran arkadaşımızın ya da izleyicimizin belirttiği gibi tadlar özellikle escape genleri ya da kaçış genlerinin çevresinde bulunuyor. Bunun şöyle bir spekülasyon yapabiliriz ya da bizim düşüncemizi söyleyebilirim. Bu genlerin diğer baskı altında olan yani silent olan bölümlerden ayrılmasını ve kompartmentalize olmasını sağlayabilir tadlar özellikle. Ve bizim gördüğümüz yani polimerazın bağlanmasını da daha kolay sağladığını düşünüyorum tadların bu bölgelerde özellikle. O yüzden çalışmalarımız aslında bu tadların ve escape ya da kaçış genlerinin X inaktivasyonuna ya da X kromozomuna göre nerede olduğuna da bakıyoruz. Yani biraz önce gösterdiğim gibi express olduğu zaman bütün X kromozomunu bağlanıyor ve bütün kromozomu kaplıyor. Tek bundan kaçış yapan genler kaçış genleri. Onların da bu tadlar yoluyla fiziksel olarak bu cloud'un dışına çıktığını biliyoruz özellikle. Ama bunlara tadlar fonksiyonel olarak burada görev yapıyor mu, yapmıyor mu? Yani tadları direkt target ederek yok etmekle buna bir cevap bulabiliriz. Ama henüz bunun tam bir cevabını bilmiyoruz. Ama kompartmentalizasyon olacağını düşünüyorum.
Çok teşekkür ederiz hocam cevabınız için. O zaman sıradaki soruyla devam ediyoruz. Plasenta gibi ekstraembriyonik bir saniye hocam, bir okumakta zor çektim. Ekstraembriyonik dokularda fare plazental hücreleri yalnızca damgalanmış paternal X inaktivasyonu sergilerken insan plasental hücreleri rastgele XSI sunar. Bu durum hayvan modeli araştırma sonuçlarının insana çevrilmesinde sınırlama yaratır mı?
Evet, Beyza'nın sorusu güzel bir soru. Çok fazla başlangıçta bu konuya girmedim. X inaktivasyonun iki tipi var. Bir tanesi imprinted X inaktivasyonu, diğeri de random X inaktivasyonu. Beyza'nın da bahsettiği gibi bu durum hem fare sisteminde hem de insan sisteminde farklılıklar gösteriyor. Tabii ki insan çalışmaları çok daha kısıtlı. Özellikle imprinting olmasından dolayı değil, sadece materyale ulaşım anlamında kısıtlı. Ama tabii bunu şu andaki çalışmalar özellikle insan kök hücreleri kullanarak ya da epi tipi hücreler kullanarak kompanse edebiliyoruz. O yüzden iki sistemin de paralel olarak kullanılması bence şu anda mümkün ve bir kısıtlama, ya tabii ki imprinting çok kolay maintain edilen bir proses değil. O yüzden hücreleri izole ettiğinizde imprinting'i kaybediyor olabilirsiniz. Hücreleri culture ettiğinizde de kaybediyor olabilirsiniz. O yüzden imprinting'i culture sisteminde çalışmak çok daha zor. Daha çok hani organ ya da organizma düzeyinde çalışmak daha uygun. O yüzden tabii ki insan çalışmaları daha kısıtlı ve daha güç ilerliyor.
Çok teşekkür ederiz hocam tekrardan cevabınız için. Sıradaki soruyla devam ediyorum o halde. X-ist eksikliği ya da delesyonunun tümörogenezdeki olası etkileri nelerdir?
Biraz önce bahsettiğim gibi sanırım tümörgenez denerek insan tümöründeki etkileri burada sorulmak istenmiş. Şöyle bir cevap verebilirim. Şimdiye kadar yapılan, daha doğrusu biliyorsunuz insan hücrelerinde eksin aktivasyonu ilk gözlendiğinde 1955-60 yılları civarında yapılıyor. Bunlar da Moore tarafından yapılmış çalışmalar ve Moore yani bar body olarak da nitelendiriliyor eksin inaktif X kromozomu ve ilk gözlemledikleri belirli kanser tiplerinde inaktif X kromozomunun ya da bu bar body'nin yok olması ya da daha fazla X kromozomunun bulunması. Daha sonraki çalışmalar özellikle göğüs kanserinde, overi kanserinde ve kan kanserlerinde gözleniyor ki direkt olarak olmasa da epigenetik olarak bu hücrelerde X kromozomuna bağlı değişiklikler gözlemleniyor. Bizim yani şu anda aslında X kromozomunun reaktivasyonunun direkt olarak tümöre bağlantısı konusunu biz araştırmaya çalışıyoruz. Ki farede yaptığımız çalışmalar direkt olarak insan çalışmalarına nasıl etkiler diye sorarsanız, mesela MPN-MDS'i gözlemliyoruz exist'i yok ettiğimizde fare modelinde. Ama mesela insan modelinde MPN-MDS'e baktığınızda acaba hani dişiler yani dişilerde daha mı fazla gözleniyor diye sorarsanız hayır, daha çok erkeklerde gözlenen bir rahatsızlık. Ama tabii bunun şöyle bir dezavantajı da var çalışmaların. MPN-MDS yaşla, daha doğrusu ileri yaşla bağlantılı bir rahatsızlık. O yüzden erken yaşta elimizde çok fazla data yok. Çünkü genelde hastalar bunu 50-60 yaşından sonra görüyorlar. Çünkü çok yavaş ilerleyen bir hastalık ve dediğim gibi yaşa bağlı olarak. O yüzden şu andaki çalışmalarımız aslında kolaborasyon yapıyoruz. Nelson Chau'nun ve Luigi Racciopi'nin labıyla Duke'de. Özellikle MPN-MDS hastalarında eksin aktivasyonu yok olmasının ne sonuçlar verdiğine bakmaya çalışıyoruz. Umarım bir dahaki sefere sizlerle birlikte olduğumda onun sonuçlarını da sizlerle paylaşabilirim.
Çok teşekkür ederiz hocam cevabınız için. Sıradaki soruyla devam edelim o halde. Meme kanseri %1 oranında da olsa erkeklerde de görülüyor. X-ist metilasyon profili tümörogenezde bozulmuş olabilir mi? Olası etkileri nasıl belirleyebiliriz?
Güzel bir soru. Bu gerçekten doğru Efekan. Çünkü özellikle erkeklerde ve ekstra X kromozomu olan erkeklerde meme kanserleri yani meme kanserlerinin olma olasılığı daha yüksek. Bu da ilk belirlenen fenotiplerden bir tanesi ve benim de exist'in dozajının ya da X kromozomun dozajının kansere nasıl bağlı olduğunu ya da faktörlerin neler olabileceğini araştırmama sebep olan gözlemlerden bir tanesi. Dediğim gibi exist'in metilasyonu ve diğer X-link genlerin metilasyonu değişiyor. Meme kanserinde ve bunun olası etkilerini belirlemek için bir kolaborasyon yapıyoruz şu anda. Farklı meme kanseri hücrelerindeki exist'in ve X-linked genlerin ekspresyonuna bakıp, yani düzeylerine bakıp metilasyona karşılaştırıp bunların three dimensional sphere assayiyle proliferationlarındaki değişiklik, differentiation'larındaki değişiklik neler, bunları gözlemliyoruz ki geçtiğimiz sene ya da ondan önceki sene Edith Hurd'un grubu Heidelberg'de gösterdi ki eğer meme kanserine sebep olan özellikle stem hücrelerinde egzisti yok ettiğinizde hücrelerin gelişiminde büyük değişiklikler oluyor. Yani egzistle direkt meme kanseri arasında bir kozal bağlantı var.
Anladım hocam. Çok teşekkür ederiz cevabınız için. Sıradaki soruya geçiyorum o halde. X kromozom inaktivasyonu sırasında ortak X-sist aracılı mekanizmaları ortaya çıkarmak için hangi modeller kullanılabilir? Fare modelleri tüm kanser türleri çalışmaları için standartlaştırılabilir mi?
Şimdi ortak exist aracılı mekanizmalar dendiğinde şöyle anlatayım. Exist'in altında işleyen farklı mekanizmalar olduğunu tahmin ediyoruz. Bunlardan bir tanesi şöyle komplikasyonu şöyle anlatabilirim. Exist'e bağlanan yaklaşık 400'den fazla protein olduğu biliniyor. Ama bu proteinlerden belki 10 tanesini, belki 15 tanesini karakterize etmiş durumda şu andaki çalışmalar. Tabii ki bu hızla devam ediyor ama exist hem RNA'ya bağlanıyor hem kromatine bağlanıyor. Ve exist'e bağlanan proteinler hem kromatin bağlanan proteinler hem DNA'ya bağlanan proteinler. O yüzden proteinleri karakterize etmek biraz güç. O yüzden aslında bizim amacımız ilk önce exist'in yok olduğunda hangi genler farklılık gösteriyor ve bu genlerin karakteristiklerine göre mekanizmaları bulabilir miyiz? O yüzden de aslında transcription faktörlere yoğunluk vermeye çalışıyoruz ki exist'in altında çalışan ya da alt kademelerinde çalışan transcription faktörleri bulup onların cross reference ettiği ya da cross talk ettiği diğer signaling mekanizmaları yani belirleyebilelim. Sanırım ikinci sorusu yani sorunun devamında hangi modeller tüm kanser türleri şu anda standartize edilmiş bir model yok. Yalnız exist daha önce de belirtildiğim gibi biz hematopoietik hücrelerde deletion oluşturduk. Benim daha önceki Ginny'nin laboratuvarında yaptığım çalışmalarda B hücrelerini target etmiştik. Epitel hücrelerde target ettik. Intestine'de ve brain'de ve kidney'de target edildi. O yüzden aslında soru şu anda oldukça açık. Ama diğer model yani hematopoietik sistem dışındaki sistemlerde görüyoruz ki exist'i yok ettiğinizde organlar gayet güzel tolere ediyor exist'in yok olmasına. Yani bir şekilde bir compensation var. O yüzden de düşüncemiz zaten bu exist'e bağlı olan hem X-linked genlerindeki değişiklik hem de transcription factor'daki değişiklikler özellikle hücrelerin bölünme karakterlerine bağlı olarak gerçekleşiyor. Yani öyle bir şey ki düşünün biz exist'i özellikle hematopoietik sistemde embriyonik 10,5. günde yok ediyoruz. Ama fenotip 3. ayda oluşmaya başlıyor. Yani 3. ayda biz kanserin ilk ebarelerini görmeye başlıyoruz. O yüzden oldukça uzun bir süreç. Bizim de düşüncemiz aslında bu bir hiyerarkiyal bir olay ve exist'i yok ettiğimizde belirli genler upregüle oluyor ve zaman içinde hücreler divide etmeye devam ettikçe bu X-linked genlerinin yani açılmaya başlayan ya da overexpress olan genlerin miktarları artıyor. O yüzden de daha da yani popcorn efekt gibi. Yani başlıyorsunuz açmaya ve üzerindeki bu silencing efekt zaman içinde ve disease daha da progress ettikçe daha da kötüleşmeye başlıyor ve daha da severe bir hale alıyor. O yüzden çalıştığınız sistemin hangi hücreleri kullandığınızın, onların division rate'inin çok önemi olduğunu düşünüyorum.
Çok teşekkürler hocam cevabınız için. Sıradaki soruyla devam ediyoruz o halde. Pardon hocam, ufak bir X kromozomu üzerinde farklı fakat evrimsel olarak korunmuş dozaj telafilerine sahip ve tümörogenezde cinsiyete bağlı değişiklikler gösteren gen grupları olabilir mi?
Olabilir. Özellikle bu çalışmalar gene şöyle anlatayım. X'in aktivasyonunda rol alan tek long non-coding RNA exist değil. Exist'le birlikte aktive olan ve ya da suppress edilen bir sürü long non-coding RNA var ve bunların her biri belirli stage'ler, yani belirli evrelerde, X'in aktivasyonu evrelerinde ekspresyonlar ekspres edilmeleri değişiklik gösteriyor. Ama soruya gelecek olursak bu long non-coding RNA'lerin çoğu da evrimsel olarak korunmuş, yani en azından sekansları farklı organizmalara baktığınızda evrimsel olarak korunduğunu gözlüyoruz. O yüzden proses dedikleri gibi evrimsel olarak korunmuş ve mekanizmalar evrimsel olarak korunmuş olabilir. Ama bu demek değil ki mesela fare sistemine baktığınızda 12 ile 13 ya da en fazla 18 tane escape geni görürken insan sistemine baktığınızda neredeyse X-linked genlerin %15'i, ki bunlar P armının ucunda toplanmış durumdalar, %15'i neredeyse X'in aktivasyonundan escape gösteriyor. Ve bu P armı da Y kromozomuna homolog olduğu için farklı mekanizmalar kontrolü altında olabilir. O yüzden her ne kadar evrimsel olarak benzerlik gösterseler de ya da korunsalar da mekanizmalar farklı organizmalarda değişiklik gösterecektir.
Çok teşekkürler hocam cevabınız için. Bu bizim son sorumuzdu. Bu kutlu, mutlu ve gurur dolu bu hafta sonunda değerli vaktinizi bize ayırdığınız, bu bilgi dolu bu konuşma için hem kendim hem ekip arkadaşlarım hem de değerli katılımcılarımız adına sizlere çok teşekkür ediyorum.
Ben çok teşekkür ederim Sude'cim. Tekrar hepimize mutlu, güzel ve nice Cumhuriyet bayramları diliyorum. Cumhuriyetimiz kutlu olsun. Diğer katılımcılar gibi ben de Atatürk'ün bize sunduğu eğitim sistemiyle yetişmiş bir genç olarak geldim Amerika'ya. Ve sizlerin de bu parlak yüzlerinizi görmek ve sizin heyecanınızı bugün de yaşamış olmak ve beni de bu güzel organizasyona kattığınız için çok teşekkür ederim. İyi akşamlar dilerim hepinize.
Teşekkür ederiz hocam. Sizlere sağlıklı günler diliyorum.
Ben de teşekkür ederim.